Good morning, good afternoon, good evening again, everybody. Welcome to the last of the plenary talks in Biodiversity Genomics 2020. It's my great pleasure to introduce a colleague of mine, Mara Lawniczak. So Mara is a group leader at the Wellcome Trust Sanger Institute, where she sits between two programs in the Institute, one to do with parasites and microbes, where she works on malaria and mosquitoes, and the other in Tree of Life, where she's helping drive through the innovations and the changes we need to start sequencing all life in Britain and Ireland, and then the whole world at scale. So Mara started out her science life as a population biologist and has for many years worked on malaria, both malaria parasites and the vectors that transmit them. And so today she's going to talk to us about her current work, most of it unpublished, on the biology and genomics of malaria vectors and what this work she's doing promises for the future. So I now hand over to Mara.
Hello, my name is Mara Lawniczak, and I think Mark will have just introduced me, so thank you, Mark. A reference genome transforms our ability to study an organism, and this fact really makes projects like the Darwin Tree of Life so exciting, because in the next few years, these projects will catapult so many species, thousands of species, into genome-informed ecological and evolutionary research. Today, I'm going to talk about that journey for Anopheles mosquitoes. Anopheles mosquitoes exist everywhere. They are, except for Antarctica, I think, they are there are about 500 described species, and about 10% of these are able to transmit human malaria. So these species are also really well known for their ability to share alleles. They're poster children for incomplete reproductive isolation. And I'm going to focus today on species from Africa, and here you can see schematics of their geographic distributions. In yellow and red and blue are Arabiensis and Kaluzi and Gambi, and these are all species from the Gambi complex that are major malaria vectors. And in pink is Funestus, where I'll talk a little bit about this species as well. It's much less well studied, but it is also a major vector. And these four species are major vectors, partially because they have these very wide geographic ranges, so they're transmitting malaria through this entire part of Africa, but also because they are highly anthropophilic and prefer to feed on humans. As you know, Africa still has a huge burden of malaria, where there's more than 200 million cases every year and over 400,000 deaths, many of which are in children under the age of 5. The Gambi complex is notorious for this, for these porous species boundaries, and that has made really understanding its historic speciation events more complex and challenging, because so much of the genome can move between species. So this phylogeny is based just on part of the X chromosome, where it seems that there's been a little bit less hybridization, introgression happening between species. And although this is the group of Anopheles mosquitoes that are the most well studied, this Gambi complex, new discoveries are being made regularly. In the last 10 years, new species have been identified and named. And there are new populations like Gundry and Tengrella that look like they are also probably going to be new species, but more work is required. The Anopheles gambi has been in a privileged position of having a reference genome for a long time. So in 2002, that's 18 years ago, this is the schematic of the reference genome. Like many flies or Diptera, there are about 3 chromosomes, and you can see that there's a heterochromatic region on these chromosomes. Those pieces are very poorly assembled still, and PEST is the name of the genome that we tend to use. It's our standard Gambi genome. It was sequenced before we really understood Gambi and Kaluzi as separate species, and so it's actually a mix of these 2 species. And still, to this date, there remains a large chunk of the genome that we call the unknown, which is just a bunch of contigs that nobody knows where they go. Now, what do we know about Gambi and Kaluzi speciation and reproductive isolation? Well, when people are studying speciation, they try to kind of classify whether there is evidence of prezygotic or postzygotic isolation. So does something go wrong during mating or before fertilization, or is something wrong in the hybrid. And as far as Gambi and Kalutsi go, which historically were called S and M, there does appear to be a little bit of prezygotic isolation. So Kalutsi, or M, tends to like to lay its eggs in these man-made rice fields, whereas Gambi tends to like to lay its eggs in these temporary rain-dependent puddles. And because of that, they also have different developmental rates. Gambi is a more rapid developer, and Kalutsi tends to be able to avoid predators better, probably because it's encountering more predators in these larger bodies of water. There also appears to be a bit of behavioral isolation, but this is obviously very incomplete because there's lots of hybridization that still happens. And these, the males swarm at night, at dusk, and the place at which the cues that the males are using for Kalutsi and Gambi seem to be slightly different. And then the female comes into the swarm and mates and finds a mate, and they match their flight frequency tones. And the exact tone at which this matching occurs seems to also be slightly different between Gambi and Kalutsi. So that's further evidence for some prezygotic isolation, but this is very imperfect. As far as postzygotic isolation goes, the hybrids between these two species are completely fertile and viable. There's no evidence of anything going wrong in the zygote itself. And in terms of whether the hybrids do well in the parental niches, that's much less well studied. So 15 years ago, a paper came out which coined the term Speciation Islands on these two species, M and S. And at the time, the DNA was hybridized to a microarray from Kalutsi and from Gambi, or from M and S. And these stars indicate these regions of the genome that, based on this DNA hybridization to a microarray, looked like the only places where there was real divergence between these two kind of incipient species. And so it really seemed like, if the model became really that that ongoing interbreeding could basically homogenize the vast majority of the genome, and just these few little places probably contained the speciation islands, or the genes that contribute to reproductive isolation. So some years later, I ended up working on mosquitoes, and we sequenced the genomes of M and S to see what was different in these islands. And this was really an effort to try to pin down these speciation genes. But instead what we found, so here in blue are those speciation islands, instead what we found is actually there was a lot of divergence across the entire genome. Each of these arms shows a lot of red, and those are fixed differences between two colonies that were established from Mali, from really the same region. And although the coverage was very low, and these are probably in many cases not true fixed differences, it does indicate that there was divergence happening between Kalutsi and Gambi at a much wider scale than was appreciated from the microarray research. And this research and other research led to the naming of these two forms, M and S, as Kalutsi and Gambi. Remarkably, actually, in the last few years, further interesting biology has been discovered about these. And it looks like Kalutsi, actually, in this diagram here, of course, these mosquitoes are dependent on water for their ability to breed. So during the long dry seasons, they disappear. But Kalutsi has a little bit of a peak in some places where there might have been a bit of rain, and it appears to do this little blip of reproduction in the dry season. And then as soon as the rains come, it's right there. It appears straight away. Whereas Gambi takes a very long time to reappear after the rains come. And this looks to be like very clear support for Kalutsi sticking around and estivating, and Gambi returning through long distance migration, which is actually very common in insects, but really really understudied. So the same group that discovered that did some follow-up work where they released these helium balloons with these kind of sticky papers up into the sky, very high, 50 to 300 meters in the sky. And they did this over years, and they caught over 400,000 insects. Only 200 of these were Anopheles, and 23 of those were actually Anopheles Kalutsi, which was a little bit surprising considering what I just told you, where we think that actually it's Gambi that does the long distance migration. So this group is still working on this, but it looks like perhaps Kalutsi actually has a mixed estivation and migration strategy. And we are actually trying to sequence the genomes of these mosquitoes that were caught on the balloons, and that'll be exciting to see if we can pin down where they came from. And that's related to this Anopheles Gambi 1000 Genomes project, which I'm going to talk about briefly here. And I'm going to talk about the phase 1 data in which we sequenced the genomes to 30x coverage using Illumina of 765 wild-caught Anopheles Mosquitoes in these red dot populations and Gambi in the blue dot populations, in order to start to really understand what was population structure like and how much gene flow is occurring and how much these mosquitoes actually move around. This is a huge project that involves tons of people, lots of people who are analyzing the data and who are contributing the specimens, and I won't name them all, they're here. This is to remember to say that we released the data publicly. In fact, we released the phase 2 data, which includes some more specimens of Mosquitoes and Gambi from more populations 3 years ago, but that paper actually just came out last week. And our phase 3 data, which doubles the number of specimens we've sequenced, again, each to 30x, and includes the new species, Anopheles arabiensis, will be released later this year. You can interact with the data. I've left a tiny URL up here, 1,000 mosquitoes. So this is a wonderful web browser called Panoptes, and this is just zooming in on 600 bases here of 100 specimens from Angola that are Mosquitoes, 100 specimens that are Mosquitoes from Burkina, and 100 specimens that are Gambi from Burkina. And each of these blue areas is a place in which these 100 genomes completely match the reference genome, the reference genome. Each of these green triangles up here is a place where there's a SNP, and you can also get a sense of that from all the green dots and red dots here. Every green dot is a place where a mosquito is a heterozygote, and every red place is a place where it's fixed for the alternate allele. Both haplotypes suggest that it's diverged from the reference genome. So I hope what this gets across is there's a huge amount of variation segregating in these populations. And that's also, you can see that also here, where we were able to look at about 140 megabases of the 250 megabase genome, just because a lot of it is heterochromatic. We're not super confident that we can actually call variants there. And across these 140 megabases, we discovered 52 million SNPs. And each individual on average has between 2 and 3 million SNPs, which is pretty remarkable. I won't talk about gene drive here, but gene drive is a huge area of research in Anopheles in terms of vector control. And basically the idea that you could release mosquitoes that would either replace a population and make them resistant to parasites or suppress a population and cause it to crash. These are based on CRISPR approaches. And these levels of variation are very problematic for drive, but there's a real good awareness now of how much variation is there. And a lot of research is really focused on places where there's very high levels of conservation now. But a few years ago, this was a real guide RNA cut site that looked promising, and just segregating in these 765 individuals were 8 SNPs in that small little region. So you can get it, you have a sense that there's a huge amount of variation, and that resistance alleles are basically already present in the population before any kind of gene drive implementation would even be released. The amount of the sort of patterns of relatedness across the genome really vary depending on where you look. So here in pink, in the centromeric regions of each of the chromosomes, you can see that Mosquitoes and Gambi are pretty different from each other. And so it's really the species that are driving that divergence. Whereas in the sort of turquoise color, it's just about geographic distance really driving the levels of relatedness. And then in a couple of places in the genome, it's really these inversions that cause which karyotype the species is, the specimen is carrying is really driving what the pattern of relatedness looks like across the populations. So another thing that's really interesting to look at in these organisms is insecticide resistance, because these are just absolutely hammered by our attempts to try to eradicate them really through insecticides. And so when these interventions are thrown at the mosquitoes constantly and for many, many years now, it pushes them down one of two paths. It either gets rid of them fully or it sets them down a path of resistance. And the two main vector control approaches that are used in Africa are insecticide-treated bed nets and indoor residual spraying. And you can see here, this bed net is protecting the children sleeping under it, but it's not just a physical barrier. It's actually, the net is impregnated with insecticide. So when the mosquito is trying to get into the net to bite the children who are sleeping under it, it's given a dose of insecticides by landing on that net regularly. And the same thing happens with this IRS, where actually typically after a mosquito has taken a blood meal from a sleeping person inside, it goes to rest on the wall and then gets a dose of these insecticides. So in a highly cited paper from 5 years ago, they estimated the number of malaria cases that had been averted due to bed nets, and it was huge. So millions of malaria cases. Bed nets are basically doing a great job. Of course, there's lots more to do. And one of the biggest problems is that resistance is everywhere. There's resistance to all 4 classes of insecticides that are used for the indoor residual spraying, and pyrethroids, which are the only approved type of insecticide for bed nets. There's massive resistance everywhere. I'm going to talk a little bit about target site resistance here. So this is a schematic of the voltage gated sodium channel gene, and this is a gene that's an essential component of the insect nervous system. And this amino acid here at position 995 in the susceptible mosquitoes is a leucine, and then there are 2 mutations that can happen at that same amino acid. One changes it to a serine and one changes it to a phenylalanine, and that confers a lot of resistance to DDT and pyrethroid binding in this channel. There's another mutation at amino acid 1570 that even increases this resistance further. And there's been a lot of research over the last decades on typing this mutation in particular in individuals to estimate the proportion of mosquitoes in populations that are resistant. And it looked primarily like the phenylalanine mutation was most prevalent in West Africa, and the serine mutation was most prevalent in East Africa, and that these 2 mutations had arisen once or twice, and that they were sweeping across the populations. But in fact, the real story is much more complicated and dynamic than that. Here are those 2 mutations, the 995S and the 995F, and these black bars indicate haplotypes that contain that serine or phenylalanine mutation. And what you can see is that they are interspersed by susceptible backgrounds. And so what's really happening here is that the resistance mutation is arising repeatedly on a susceptible background and sweeping in that population. And not only sweeping in that population, as you can see by these blocks of color from Gabon and from Uganda, but also intermingling between the species. So this red and blue intermingling, it's arising in one species and then being transmitted to the next quite frequently. There's another type of insecticide resistance that's referred to as metabolic resistance. And instead of a target site and an amino acid change, this is really believed to be driven by copy number variation at detoxification enzymes like cytochrome P450s and glutathione transferases. These probably help catabolize the insecticide before it reaches the target. So we also looked for evidence of gene duplication variation in the genomes, and the same exact story is present. Really remarkably, this cluster of cytochrome P450s, it looks to be like there are 15 independent major copy number variants that are present in the populations, all covering the same set of genes, which are believed to metabolize pyrethroids. So these 2 stories are just 2 examples. There's many in the genome at other important insecticide resistance loci, where it looks like resistance is arising regularly and moving locally, but also between species and across great geographic distances. And this is, almost certainly due to the fact that we are constantly hammering these species with insecticides. So it's perhaps not so surprising retrospectively to see so many independent arisals of these mutations. What we really want to work on now is to try to understand much more spatial and temporal detail of how these mutations arise and spread. And so we have started on a project that we call the Vector Observatory at Sanger, in which over the next 5 years, we expect to sequence in a much more strategically oriented regular sampling in the same locations over time. So we have about 100 sites that we will be working in, where we'll be sampling every few months regularly over the course of the years. And we will probably sequence on the order of 50,000 individual mosquitoes from all 4 of these species to really understand in a much more dynamic sense, where we also have a sense of what the vector control implementations are. How are these mosquitoes responding? How are alleles moving between populations? As part of laying that foundation, we've started an Anopheles Mosquitoes population genomics project I told you that our phase 3 data covers Gambicolutzi and arabiensis, so we've also got this other study in which partners listed here have shared samples with us from these sites across Africa of Funestus. And we've sequenced 1,000 individuals now, and I won't show you any data from that because we're still trying to put together a better Funestus reference genome, which will come up later in the talk. But we should have a great foundation from this project with these partners of understanding what Funestus genome structure and allele sharing looks like across Africa. Not only do we want to start regularly sampling now, but we also want to understand what did mosquito genomes look like before insecticides, and that's the foundation of a project that we call Project Neandersquito. You might wonder where we would get access to historic specimens, but actually there are 511 drawers that look just like this. Each one of these pins is holding a mosquito collected a long time ago at the Natural History Museum in London, where there are 3/4 of the world's known mosquito species. This collection is curated by a woman called Erica McAllister, who's a great science communicator and has a couple of wonderful books I highly recommend. We are primarily focused on looking back in time at the Funestus and Gambi groups, given our modern day genomic data. And this actual box has just the Gambi. These are just Gambi specimens. Here's a picture of a mosquito from that collection. You can hardly see it, but the tag says 1919, so it was collected 101 years ago. And this female here has a blood meal contained in her abdomen. So we are optimistic that not only will we be able to look at the genomes of the mosquitoes, but also understand who that mosquito bit through the host DNA, and if that person was infected with malaria. So Petra in my group has been working, and I have an asterisk here to note that she's done all these drawings. She's been working to develop a protocol with the museum where we can non-destructively extract the DNA from these specimens, because of course these are precious collections that the museum is totally not interested in having us destroy. And so she's worked out a protocol that we think is going pretty well, where we take the mosquitoes, we put them in a box with some water basically to rehydrate them so they're not quite so dry and crumbly. Then they get submerged in the lysis buffer that releases their DNA. And then they go through this process of what we call rinsing and refluffing, which is a critical point drying that basically puffs the mosquito back up. Sometimes it looks even better than it did when we got it. This lysis buffer, this non-destructive lysis buffer, is also working really well for us for modern day samples, and that'll come up later in my talk. One of the most fun aspects of this project is actually the fact that you can go back into archives of people who were traveling around Africa and trying to study different species of Anopheles mosquitoes. And so here are the boxes of a man's collection from 1936. This is a guy called Herbert Leeson who went to Africa to study Anopheles funestus. And in these boxes are all sorts of interesting things, drawings and maps. Here's a menu in which they were on the train and there were glazed sheep tongues available for lunch. There's also film footage of this trip in 1936. And this picture over here is of the actual plane. They took some film footage while they were flying. They flew from Croydon in the UK to Entebbe in Uganda. It took them 5 days. They were flying pretty low and they have footage of flying over Africa. And this is the opening slide to the film. And it says, the story of an expedition to East Africa led by Mr. H.S. Leeson to investigate the bionomics of Anopheles funestus, a malaria-carrying mosquito. And Leeson was accompanied by this guy called David Gillett, who narrates this film many, many years later. I think in the 90s it's narrated. And it's really enjoyable. So this is the vehicle they traveled around in Africa with. And here is Leeson on the back of that vehicle. You can't see it, but there's a microscope here. And he's looking at some mosquitoes that he's collected. They were accompanied on this flight by this letter here, in which there was some, I don't know who it was to, but it was basically to convince somebody to keep Gillett on to be an assistant in another study. And it says some funny stuff like his scholastic record was so bad, but in every other respect, he's a very unusual and able person, one of those born naturalists, and to speak snobbishly, a gentleman. Here is Leeson using a soup ladle, which is an essential piece of vector biology equipment to catch larvae swimming around in water. Here's some drawings from a collection of them trying to work out different wing patterns. And This Anopheles funestus, Anopheles leesoni, which is named after Leeson, obviously, and Anopheles X, which if you work on Anopheles mosquitoes, this is a very near and dear to your heart term, because it's often, you're like, what the heck is this thing, Anopheles X? And this is just to say, 80 years later, we were in Uganda, possibly in the same places where they were collecting, and the soup ladle remains an essential piece of equipment to catch larvae. Here's my daughter looking at some of the larvae we collected. So as I mentioned, we're still trying to get the reference genome into good shape, so we haven't looked at the nuclear DNA for this, but here's the mitochondrial results from in here in red, all these specimens of funestus that we've now got genome sequence data from Leeson's collection. In these, the story I was just telling you about, these are the actual mosquitoes that they collected, which we've now sequenced the genomes from. And you can see that the red is interspersed in both these lineages, and these lineages are not well studied, but probably are different species. So we're excited to look at the nuclear DNA for this. And in some, this is just to give you a sense of the kind of coverage we have. So we have really good coverage for a large portion of the DNA, which is very exciting for mosquitoes from 1936. Because I can't show you anything about funestus nuclear genome, I'm going to show you instead we've also sequenced maybe 50 or so mosquitoes from the Gambi complex. And here on the left is that 995 mutation, 995 amino acid in the VGSC gene. And where you see red or green, that means that individual is resistant or heterozygous for resistance at the VGSC gene. And here are the historic specimens, and you can see it's all blue, it's all susceptible. So even as late as 88 in Kenya, we are not seeing any evidence of resistance in the specimens we've sequenced so far. So we are in a really fortunate position in Anopheles to have lots of reference genomes. These are the species that we have genomes for so far. And pest is here, and you can see the number of scaffolds is unfortunately still in the thousands for most of the species. And here's pest with just 7 scaffolds. But of course, that's a little bit misleading because there are more than 20,000 gaps in the genome. So some of the problems, a lot of this has been generated by short-read data, but another big problem for assembling Anopheles genomes is that until recently, mosquito genomes were using pooled DNA. So we are, a couple years ago, we released a paper in which we worked with PacBio to use just DNA from a single mosquito to generate a reference genome. And in the process of doing that, before that actually happened, we were already in the process of talking to a bunch of partners listed here who were willing to kind of go out into the field, collect a wild female who had mated, bring her back to the lab and rear her offspring. And so we were going to try this kind of pooling approach. And so all these partners had collected these mosquitoes and bred them for us. So they had mothers and offspring. In the end, we gave up on the pooling approach and we're just using single mosquitoes. And I'm going to talk you through one example of this. So this is work that Harriet and Katerina and Alex have all contributed to in my group. And this is Anopheles funestus mother collected in Gabon and the brood that she came along with. And these are the partners that contributed that. So these mosquitoes were shipped to us. We extracted them using our high molecular weight extraction protocol. And this is the kind of pattern that we would see from these mosquitoes, where we've got some decent high molecular weight there, DNA there. And from this particular female, we had 306 nanograms from the extraction. And we called that a high molecular weight band plus a smear. Then we sheared that DNA, which left us with 180 nanograms. And then we created the PacBio CCS or Hi-Fi library from that. So there was a 40 nanogram library available to sequence with about 12 kb of the average size there. You can see this is the library. And we multiplexed this individual on an 8M cell with another individual. The GenomeScope profile estimated that the genome was about 234 megabases and a heterozygosity rate of about 1.4%. We got about 870,000 reads from this mosquito, including 8.1 gigabases of data and about a 9 kb typical size that we had lots of CCS coverage for. So how does it assemble? We decided to test 3 assemblers in this process: HiCanoe, HiPhiasm, and IPA. And here is the high C data from a sibling individual, which is only 17x step. And this is pre-scaffolding. So this just gives you a sense of the contig sizes that come straight off of the CCS data. And I should mention that for Funestus, there's a lot of, most of the other samples I'll talk about, we have many fewer contigs than this. For Funestus, we have a lot of contigs from these because there's some challenges around rDNA that seem to be falling into lots of small contigs. But the results were really impressive, and we chose to proceed with the high chiasm assembly as that it looked the best. It had an N50 contig size of 26 megabases. And even, you know, one of the most exciting things that still is really kind of not fully appreciated is that these are really kind of 2-for-1 genomes, where you're really getting that we focus on this haploid primary assembly, but there's a whole nother genome there from the other parent. And this is exciting because even the haplotypes are very large pieces. So here's the result from the scaffolding using SALSA2. And we are going to gather some more high C data, but I think we're in a pretty good position. We're going to have a pretty nice Funestus genome from this. And here it is compared to the best current Funestus assembly, which was from a different part of Africa. So we know that there should be some inversions, and you can see those here. So we think that from a single mosquito, we've got a very good Funestus assembly now, and we have multiple different Funestus assemblies from multiple different mosquitoes, because we'd like to move into this era of kind of long-read population genomics and pan-genomes. One of the things that has been really problematic for getting these samples to us is how expensive it is. So just to ship 10 mosquitoes from Cameroon to Hingston was more than 4,000 pounds. Which we did not want to spend. So we started working out a protocol. Actually, I should also mention, not only is it expensive, but even when you do fork out the money, sometimes the dry ice evaporates. And when it does, here's a set of extractions of these samples that we received from Kenya, where the dry ice had all evaporated. And you can see all the DNA pretty much is less than 10 kb, so pretty useless for what we want to do. So we've been working out a process for trying to get mosquitoes to be shipped to us at room temperature and avoid this cold chain. And here is a result to show that I think this is very achievable for insects anyway. So we did a mock experiment in which we collected mosquitoes, we put them at a -80 for a few weeks in these different storage solutions. And then we mocked shipping, where we took them out of that storage solution and left them at room temperature for a week. And you can see this kind of stepwise function where there's an improvement in the molecular weight in the second of each of these colors. And the difference there is this SQ is letting you know that we actually gently squish the mosquito. So we put in an Eppendorf with 100% ethanol, for example, and then gently squished it with one of those blue plastic pestles. And not so much that it was decimated, but actually just enough so that you compromise the cuticle and the preservative can penetrate more easily. And you can see that in almost every case, that really makes a difference to this long room temperature week of room temperature storage in ethanol, where we're still getting great high molecular weight DNA. So Sandrine from Cameroon, instead of spending 4,000 pounds on our shipment from mosquitoes from Cameroon, she collected these broods for us, squished them lightly, and then shipped them off to us. They ended up taking 7 days to get to us, in which they were at room temperature the whole time. And I want to make sure that it's clear that this is not a perfect system. She sent us a mother and 5 offspring, and we extracted DNA from most of the offspring. And in both cases, both for Muschetti and for Nili, these are also major vectors in Africa that have no genomic resources so far. We were only able to find one mosquito where we got 200 nanograms from these. And this is the profile of the DNA that resulted from one of those mosquitoes. So we've now sequenced Nili and Muschetti, and in spite of this low starting material, we've got really good coverage. Both of these specimens were males. We think we're going to get great genomes out of these. And in fact, we've done this now for 10 different species where we've got really good PacBio coverage. We're working on the high C now from sibling specimens. We have a few specimens that have failed. This is just to give you some stats on these are all wild-caught mosquitoes or their offspring. The DNA quants range from 193 to 660 nanograms, And the libraries, the final libraries that were successful here range from 25 to 89 nanograms. And the thing that's in common here among these specimens that failed to sequence on PacBio, in spite of generating what looked like fine libraries, is that they were squished in 80% ethanol. They were only at room temperature for 2 days. So we think, we haven't studied this extensively, but we think that the ethanol percentage might matter. So once you have a lot of reference genomes, it's possible to do all sorts of really cool things like generate new tools for surveillance. And this is a schematic of a project that's been led in my group by Alex and Petra, in which we've taken the available reference genomes and we've identified 62 loci across the genomes that are phylogenetically informative, 200 base pieces of the genome that are phylogenetically informative, flanked by very highly conserved primer binding sites. And what this allows you to do is sequence targeted amplicon sequencing of these 62 loci from any anopheline that you push through. And that data then gives you a sense of what that specimen is. So here is the result for 40 different species that we've pushed through this panel now. And you can see this is the recovery of the amplicons. So for the Gambi complex, for example, we get great recovery. That's probably because most of the genomes that exist are from that group. But even from things that are very distantly related, we're getting more than half of the amplicons generating informative data. And one of the most exciting things about this project is that it makes it possible to do this at huge scale. So on a single MySeq run, we can run this on 1,000 specimens. And this is really going to be something that will be great to start seeing running in Africa, where researchers who have access to a MySeq can now do much larger scale vector identification. So we're working on creating the very good reference library so that it can be kind of an automated approach of submitting the MySeq run and you get the sort of information back of what species the best guess is for what that individual is. On top of that, so here's a species tree, and you can see on the left, this is the data from the amplicon, from the targeted amplicon sequencing. This is the estimated phylogenetic relationships there. And on the right is the tree from the reference genomes, and these are very similar in their estimates of relatedness. And here we've done an in silico analysis of the Anopheles Gambi 1,000 genomes data, where we pretended that we basically PCR amplified up all those individuals, and we can see that it works very well to differentiate Kaluzi and Gambi too. I want to mention as well, I think I forgot to say this, but there are also 2 amplicons in this panel that target the Plasmodium parasite. So not only is it telling you which species it is and what kind of population structure exists within the species when you have many samples from within a species, but it's also telling you whether that sample was infected with Plasmodium, and if so, which species of Plasmodium. So we think this is going to be a really great tool to roll out widely across Africa and elsewhere. And in fact, over the next 5 years, we're hoping to apply this panel to 500,000 wild-caught mosquitoes to study this. We've also, I mentioned in the Neandersquito project that we've got this lysis buffer approach, which is able to non-destructively extract DNA. And that not only is it non-destructive, but it's also extremely cheap. So you just put a single mosquito in a well of a 96-well plate, and the lysis buffer extracts the DNA. You use that as your template for this targeted amplicon sequencing. And if you see something that's odd, like Anopheles X here, which I'm trying to represent, you can actually go back to the specimen and identify whether there's any morphological characteristics that help you see what makes that specimen different from another species. So in the final few moments, I just want to talk about another global project like the Earth Biogenome Project, but this one is called Bioscan, and it's led by the International Barcode of Life. And it's aiming to use targeted amplicon sequencing also, but focused on the plastid genomes at huge scale of species from across the whole world. And this is meant to do new barcoding, DNA barcoding for 10 million specimens, Symbiom barcoding for 5 million specimens, and meta barcoding for bulk specimens. You can read more about it at that link. So what is Symbiom barcoding? Well, the way that this Bioscan project works is that it's simultaneously amplifying from a single specimen all the taxa that are found within and on that specimen, as well as the specimen itself. So it's amplifying 18S and ITS for fungi and RBCL for plants and cytochrome oxidase 1 for animals. Thousands of samples, once they've gone through this PCR, amplifying those different barcodes, can be sequenced simultaneously on single PacBio runs. And through this targeted sequencing from a single specimen of a fly, for example, you can identify what that fly is and what it was eating, what it was pollinating, and whether it was infected with other parasites. So we're really excited about this global project. And over the last year, we've set up a Malaise trap in my garden. So a Malaise trap is something that actually, you can see the schematic there. An insect, a flying insect, will fly into that kind of gray area here, and there's a tent up above it that's white, and the fly will fly into it and go up, and will end up funneled into this container right here that contains, in our case, we've put in 100% ethanol. And so here's a penny from the UK, and this is just a small set of insects that would come out of this Malaise trap set up in my garden. And so what we've been doing for the last year is now plating these specimens individually into wells with a lot of different lysis buffers to test what might be giving us the best DNA. So these different lysis buffers, so here's a close-up of samples trapped from my garden, and we release the DNA into this, and we want the DNA to be future-proofed for things like the Darwin Tree of Life project. So for example, if we said, oh, we don't have this fly's genome, we can say, okay, well, we've seen it on Bioscan, we know what it is, we can go after the DNA, the specimen's still there, the DNA is still there, and is it available, is it basically quality enough for us to do long-range sequencing on? And so we're excited because it actually does look like this is possible. This is a mock Malaise trap experiment using mosquitoes, in which the mosquitoes were sitting in 100% ethanol at room temperature for the day without the squish that I mentioned before. And then we've individually plated them out into this potassium chloride buffer with proteinase K. They've been left in the fridge for 10 days, and then we do an overnight incubation at 56 degrees. And you can see that we still have some decent high molecular weight material here, which with further shearing probably is reasonable to try out CCS on. So we'll be trying that out over the course of the next month. So we are proposing also in the next 5 years to do this very large scale across the UK Malaise trapping / bioscan project, in which partners will, at long-term ecological research stations or in sites of special scientific interest, will be collecting, as part of this project, flying insect specimens using Malaise traps and plating them out for us into lysis buffer that releases their DNA but preserves their morphology. So I just want to end on the note that these two huge global visionary projects, Bioscan and the Earth Biogenome Project, have distinct but compatible goals. And over the next few years, my group will be focusing on trying to find out where we can unite those things, those two big projects for small flying insects in the UK. We want to future-proof our DNA and specimen biobanks for long-read population genomics and reference genome sequencing. And we're really excited about this Bioscan project and how this kind of space-time component is going to reveal species interactions, but also really give greater insight into the insect diversity that we are rapidly losing, and which is so critical to the health of our planet. So I just want to say, I want to plug two things, these tiny URLs here. There's a Slack workspace that's open to the community. It's called Atuda 2020. You can go get on it by going to that link, Atuda 2020. We share everything we're learning on that in terms of protocols and squishing, long-term storage, that sort of stuff. We want to work collectively across, these are big projects that we really shouldn't be spending lots of time doing the same work in parallel. When we learn something, we should share it as fast as we can. And there's a huge amount of research that's going to be required to get these data types that we need from ever smaller organisms. So how can we get high C and RNA and high molecular weight DNA all from a tiny little midge? We have a lot of work to do. So the other thing is, if you work on arthropods and you want to see particular reference genomes come out from our Darwin Tree of Life project, you can go to this link and tell us what you want to see. So with that, I will end and say thank you very much. This has been a wonderful conference, and it's been a really huge privilege to be able to tell you about the research we've been doing. The members of my team who have been really instrumental in the work I've presented today are listed up at the top here. And I also want to thank all the other people who are listed here who have been so critical to these different projects, and our funders, Wellcome and Gates Foundation. And if you are looking for a postdoc and you like the sort of data we work on, then please do get in touch. Thank you very much.
Thank you very much, Mara. Thank you very much indeed. Applause for that talk. There's some questions come in, some of them about mosquito genomics, some of them about Bioscan. We'll start off with the one from Rob Waterhouse about the mosquito genomics. Do you think it's now time to completely shelve the pest reference? And how do you think we can avoid these Frankenstein hybrids in future sequencing efforts?
Yes, I do think it's time to shelve the pest reference. I think we're close. I think we've actually got to the point now where we will, this year, I would say 2020, certainly by early 2021, be working with Vectorbase to have a really nicely annotated and assembled set of genomes, actually not just for Gambi, but for other species too, available for use. So we're still trying to generate the additional RNA-seq data that might be useful there for the same colonies that we're generating Gambi data for, and for the wild specimens as well. The other question around how do we avoid Frankensteins? I don't know that we can. I think that actually the Amplicom panel that I mentioned could be useful there, because it gives you a first little glimpse into what the sort of structure of the genome might be in terms of where these different pieces, how they relate to populations we understand. So, but this is the real biology. These hybrids reflect the real biology too. So for example, we've got hi-fi data from something that came into us as, is it Zemani or is it Kustani? Or is it a hybrid? Not sure. And we're excited to look at the data to try to figure out what is it?
Cool. So Alex Twyford asks whether or not the model of growing islands of divergence in speciation supported by large-scale genome sequencing. So rather than just a pairwise comparison, but is this pattern supported?
I think it depends on the organisms that you're looking at. So, islands of speciation or divergence have been studied in lots of different species now. For Anopheles, I think that for the Gambi-Kalutsi story, so I'll tell you my favorite hypothesis, which is I think that these species are probably actually really separated by an inversion that we haven't discovered yet. I think the X chromosome has an inversion in the paracentric heterochromatic paracentric region, and that we're going to find it with the long read sequence data. And that it will make a lot more sense as to how these two species basically share lots of stuff, but still maintain a lot of divergence in a particular place in their genome, which is really the X chromosome.
Eric Jarvis asks whether or not the segmental duplications could be due to unresolved haplotypic segments that are included in the assembly, or whether they really are duplications and deletions.
They are really duplications. So it was short read data that we used to identify those, but the scientist who worked on that and is still working on that work, Eric Lucas from Liverpool, he really carefully went through and looked at coverage there to try to make sure that these were real duplications, and he could identify breakpoints. So they're real. But it'll be really exciting to see those in long read data. And that is actually, we're trying to develop a long read insecticide resistance panel where we can amplify both haplotypes from each of these resistance individuals so that we can see these copy number variants and how they actually sit on haplotypes, not just within individuals.
And when you're assembling these genomes, it's an anonymous question, do you also assemble the symbionts and the bacteria? And how do you identify those in your assemblies and remove them, or identify them and annotate them?
Do you want to take that question, Mark? Because it will be blob toolkit. We haven't done it yet. I have a feeling, so our collaborator Diego Ayala, who has shared with us the samples from Gabon, believes that our Mouchetti sample will have Wolbachia because it's highly prevalent. And it's a difficult thing. You don't really see much Wolbachia in Anopheles actually. So we haven't looked yet. The data are fresh off this week that I was showing you. So, but it'll be exciting to see if we get Wolbachia from that specimen.
Yeah, and lots of interest in the Malaise trap and the Bioscan. So Torsten Stuck asks, how do you prevent cross-contamination between individual specimens in the Malaise trap and the alcohol in the Malaise trap, especially when it gets really full?
So I have no idea. We haven't actually done any sequencing on any Malaise trap specimens yet. And it's a question I think that will be, I think we will have contamination. I mean, you can, just when we're looking under the scope and we're separating out these little tiny flies, you can see that there's legs and wings floating around. So undoubtedly we're gonna see amplification from things that weren't actually associated with the specimen. But I think it's okay. I think we'll get, because the scale of the project globally is so big, these interactions, these symbiomes, I think a lot of the randomness will wash away and you'll see the true associations with increased sample sizes.
So a couple of questions around the general applicability of the extraction method. So would it apply to other specimens, and could you also apply it to other sorts of traps such as pitfall traps or other ways?
I think so. I mean, we haven't tried other stuff. I think it sort of depends on what your aims are. I think DNA that works for short read amplicon stuff is pretty easy at this point. It's not a real challenge. So I think, yeah, you can really mistreat those specimens. If you want the long read stuff for larger specimens, I think the ethanol might be a problem. I don't know how well it's going to penetrate, giant beetles and things like that. But particular pieces of the beetle might be penetrated well by ethanol. I think it'll depend on the question you want to go after and the specimens you're interested in. But if you want to talk about it, join a TUDA and we can share protocols and learning there. And
what about sequencing in the field with Oxford nanopore to avoid shipping difficulties?
Right now, I wouldn't do that. Not for long reads anyway. I mean, who knows what the future holds, but right now, hi-fi is just so wonderful that I don't know why you would sequence these, with specimens coming in that have this kind of molecular weight DNA, room temperature shipped from, with ethanol, like I showed for Mouchetti and Lily. These are great specimens, and hi-fi is giving great data for that. Longer term, I think there will be genomes where you really need these very long reads. Anopheles is not a great example of that, but other mosquitoes are, like Aedes. I don't know how well Aedes would do with this approach because it's much larger and highly repetitive. When you need the ultra long reads, you need really high quality DNA, then you need to do that locally, or you need to ship on dry ice. So Oxford nanopore, I think, is a great approach to use in those situations.
Yeah, and unfortunately for the other people who've asked questions, we will copy them and give them to Mara so she can answer them at her leisure, except for people who put them up as anonymous. We're out of time. So thank you again, Mara, for a lovely talk. As you'll see from the chimney breast behind Mara is probably about 10% squid or octopus in her DNA. And yesterday was International Octopus Day. So happy one day late International Octopus Day. Thank you very much for the talk. And do join us all after the break in about 5 minutes time for the closing plenary, where we will have some discussion from panel members about challenges to us as a community going forward with our grand project to sequence everything on Earth and to understand what we've done, and also to say thank you to the people who've made this possible. Okay, so we'll see you in 5 or 6 minutes.